Water quality is essential for ecosystem health and safe recreational use. In New Providence, blue holes and lakes are important aquatic environments that may be affected by microbial contamination. E. coli and coliform bacteria are commonly used as indicators of water contamination. Factors such as pH, salinity, and dissolved oxygen can influence the presence and survival of these microorganisms. This study compares blue holes and lakes to examine how water quality parameters related to microbial contamination and what these relationships mean for recreational water safety.
Between the dates of May 8th and June 1st, ACME collected and conducted water sampling at 10 different locations, equally split between blue holes and lakes around the island of New Providence. The five blue hole locations include Miami Blue Hole, Armadillo Blue Hole, Foxy Blue Hole, Frankwater Blue Hole, and the Venetian West Blue Hole. And the five locations with lakes include Coral Lake, West Lake, Lake Waterloo, Lake Orlando, and Lake Nancy. Site selection was accomplished through the cooperation of Parkland Wildlife and PBSI, and all sites were relatively accessible to the public, allowing for efficient sampling and collection to take place. The equipment used was sampling with measured pH and a YSI handheld pump, which measured dissolved oxygen and salinity. All flows were submerged underwater without touching sediment to allow for accurate readings. The difficult collections of the Venetian West Blue a modified gel apparatus was used for sampling collection. As there were multiple sampling points at each location, all samples were collected and placed together to create one mass composite sample for each site using labeling to help with identification, that was stored in large plastic bottles and were placed in cooler bags to later be diluted through a 1-to-10 ratio and tested in the lab. Probe calibration is done beforehand to ensure that there are no known deviations, in addition to the YSI contained in a water buffer to maintain accuracy. Deviations were documented. The malfunction of the pH sensor on the YSI plane leading to the usage of the hard instead, and heavy rain on the 14th led to the initial sampling of RMJ fluids defensively rescheduled by 21st.
Using t-tests, the mean pH did not differ significantly between blue holes and lakes, as the test produced a p-value of 0.785. The blue hole had a mean pH of 8.04, while the lake had a mean pH of 8.14. Dissolved oxygen concentration was generally lower in blue hole with a mean pH with a mean DOPPM of 4.57 compared to 6.97 ppm in lakes. The t-test produced a high p-value of 0.059. Salinity was also lower in blue hole with a mean of 4.45 PSU compared to 5.14 PSU. The values are not normally distributed and was therefore analyzed using a mean written test. No significant difference in salinity was detected between outside sites, formed with a pH value of 0.438. Microbial contaminations like E. coli and coliform have shown a clear difference between habitat types. All five blue holes recorded E. coli and coliform concentrations greater than 1,000 mpm per 100 milliliters. In contrast, only two lake sites, Lake Waterloo and Lake Nancy, recorded E. coli levels greater than 1,000 mpm per 100 milliliters. Similarly, only Lake Waterloo and Lake Kalani, airport lakesides, recorded coliform levels greater than 1,000 mpm per per 100 milliliters. As an example, CDC most probable number table is used to report all values. However, all values above 1,000 are simply reported as higher than 1,000 without providing an exact amount of concentration. Because many sites reach this upper limit, correlation analysis between the water quality parameters and microbial contamination cannot be reliably achieved.
After analyzing the data, we found that both glucose and lakes generally exhibit closer alkaline conditions from 6.5 to 9.40. Dissolved oxygen and salinity varied among sites, reflecting environmental and psychological factors. However, statistical analysis revealed no significant differences in dissolved oxygen or salinity. Suggests that overall, the two water body types share broadly similar characteristics. While the physical chemical characteristics were similar, the microbial results were different. Several people reported E. coli and coliform contributions to liver 1,000, and some links also showed elevated bacterial counts. These microorganisms are commonly used as indicators of liver contamination and may suggest inputs from sewage, stormwater runoff, infected animals, or other
Several limitations to be considered when interpreting the results of this study. First, the number of sample points from blue pools were limited due to their accessibility and safety constraints, which reduced the overall sample size and may have affected the comparison between the habitats. Second, the Venetian Village Blue Pool site was represented by only a single sample, limiting its ability to be compared adequately to the other sites. Finally, the microbial contamination values that were above 1,000 mpm per 100 milliliters were not provided an exact concentration number. As a result, highly contaminated sites could not be differentiated from one another. Ways that the study can be strengthened is by increasing the number of sampling sites or sample points at each site, and repeating these samplings throughout different seasons to capture the temporal variations in water quality. In addition, the use of a laboratory method capable of getting the exact microbial concentration above 1,000 mpm over 100 milliliters, so we can get a precise measurement and actually differentiate the level of E. coli at the high value site.
Correctly
This course significantly improved my skills as a field researcher by providing hands-on experience in both field sampling and laboratory analysis. In the field, I gained confidence in data collection through operating the YSI probe, and during field visits, I collected various water quality measurements, including pH, dissolved oxygen, temperature, conductivity, ORP, TDS, and salinity.
I learned some new skills through the use of the YSI and the probe, how to measure for different factors like pH, dissolved oxygen, electric conductivity, etc. So that was a good thing to learn. I never thought I would be doing water quality testing. So it was new to me, and I appreciated it very much. Is lab prep, testing, reading those charts to interpret what we see in our test kits, and it was cool to know the different levels of what is safe and what is unsafe for to be either doing recreational activities or even for consumption. It was mind blowing that some of the water